My name is Olivia Gade, and my project is the effect of hepatocellular carcinoma genotype on the tumor immune microenvironment. Immunotherapy is a form of cancer treatment that uses the body's own immune system to control and eliminate cancer cells. There are various forms of immunotherapy. My project focuses on targeted antibodies, but there are also adoptive cell transfers and cancer vaccines. Current results for immunotherapy are limited due to high rates of adverse effects and lack of research in the field. This can be improved by immune cell phenotyping of HCC tumors, which will be able to identify patients that have a higher likelihood of response to immunotherapy. This is what my project aimed to do with the beta-catenin mutant tumors and the beta-catenin wild-type tumors. Hepatocellular carcinoma, or HCC, is the most common type of liver cancer. Patients that receive this diagnosis have a 2-year survival expectancy, which is the median number, and a median survival rate of less than 50%. Most patients that receive this diagnosis suffer from cirrhosis, which can be caused by hepatitis B or hepatitis C, intake of fatty foods, or high consumption of alcohol, amongst other things. There are various forms of treatments for HCC, like radiation or chemotherapy. However, most are not curative and most are not effective for advanced stages of this disease, so researchers aim to find another treatment that can help patients. Mouse models. Human tumors that are surgically cured with early-stage HCC variants that could potentially never need systemic therapy are placed into a mouse model, as well as stem cells and progenitor cells, to create the humanized mouse model. This model visualizes the immune cells from the human stem cells and the tumor cells' interactions with each other to create an accurate model. My project takes tissue from this model and investigates the response rate of the immune cells to the tumor tissue cells. Gallage et al. Established that the liver possesses regenerative abilities, allowing hepatic recovery in the afflicted. The liver's regenerative abilities have long been known and are important in this study because although the liver is able to regenerate a lot of its tissue, it is still not able to fight off hepatocellular carcinoma on its own. Jin et al. Established that immunotherapy is a revolutionary form of cancer treatment. However, there are significant limitations to this treatment because not all tumor types are responsive to immunotherapy. Brown et al. Established that mouse models are the basis of preclinical and translational research, and the tissue from my study came from a mouse model. Lee et al. Established that the ultimate goal for immunotherapy is to effectively expand liver transplantation access while preserving pre- and post-transplant outcomes. Levet et al. Established that early surveillance programs still leave about 40 to 50% of patients that have received an early diagnosis and they still need systemic therapies. My research aims to answer the question, does hepatocellular carcinoma genotype impact the tumor immune microenvironment? I hypothesized that the beta-catenin mutant tumors would have less tumor immune infiltrating cells than beta-catenin wild-type tumors. Tissue was given to me from the PDX on slides that were processed in formalin, and I performed immunohistochemistry to visualize the immune cells. Then I put the stained cells into a Perio image scope and counted the cells in 20x field of view 5 times for each slides. Then these numbers were placed into GraphPad and Google Slides to get a data analysis and create charts. This is a visual representation of my methods. As you can see, the tissue was taken, then it was placed on slides and stained, the staining is what I did. Then the immune cells were counted, and then they were graphed. For my results, the chart on the left displays the amount of immune cells present in each section of each tumor. The chart on the right demonstrates the average immune cell infiltration for each tumor. For my more conclusive results, the chart on the left demonstrates that the beta-catenin wild-type tumors had more immune cell infiltration than the beta-catenin mutant tumors. The chart on the right further demonstrates this and shows that the majority of all of the beta-catenin wild-type tumors had more immune cell infiltration than the beta-catenin mutant tumors. In conclusion, my hypothesis was proven. The mutant tumors had a lower average immune cell infiltration than the wild-type tumors. The wild type count was 131.28 immune cells that infiltrated into the tumor tissue, and the mutant was 77.1. To compare these two values and to compare the sets of data, I used a two-tailed t-test with a p-value of significance being less than 0.05, and I found a p-value of 0.0063, which is statistically significant. This means that the comparison between the two data sets was statistically significant and the null hypothesis could be rejected. A possible reason for my findings is that mutation may lead to a decrease in production of immune signals, like cytokines or chemokines. Additionally, the mice that were used for the PDX are somewhat immunocompromised. This means that the immune cells may have not been able to infiltrate the wild type as well, the mutant as well as they were able to infiltrate the wild type. Additionally, mutant tumor is not as facile as normal tissue or wild-type tissue in maintaining homeostasis, so it may have more trouble producing the immune signals. To improve this research, data can be collected from additional tumor groups in each group. For future research, different tumor genotypes can be investigated, and new methods of counting the immune cells can work to create more accurate data. Additionally, improvements to the humanized mice model will allow more applicable results when relating the PDX to the humans. Thank you.